let's move on to the general schema of the gastrointestinal tract. Again, let's go from within outwards. First, you have what is known as the mucosa, and the mucosa consists of the lining epithelium, under which you have the loose areolar connective tissue of the lamina propria, under which you have a circular and a longitudinal muscle coat, which is belonging strictly to the mucosa, hence the name muscularis mucosae. Okay. Then we've got the submucosa, that you can see in light blue over here. Okay. In the lamina propria, you're going to have capillaries, you're going to have some lymph nodes, some lacteals, etc. Now, in the submucosa, there are submucous glands. The two important examples where submucous glands are seen is esophagus, and in the duodenum, these are known as Brunner's gland. Okay, Brunners, Brunner's gland. Please note that down. Okay, then the submucosa is also going to have vessels, but they're going to be slightly larger capillaries as compared to the lamina propria. Try to understand that whatever you eat is absorbed by the enterocyte. From there, at the basal aspect of the enterocyte, it goes into the capillary system. Then these smaller capillaries eventually join to form bigger capillaries and vessels, which will be in the submucosa. Correct? Then outside of the submucosa, you have the next layer, which is known as the muscularis externa, so that you do not confuse it with the muscularis mucosa. And the muscularis externa consists of an inner circular layer and an outer longitudinal layer. Okay, now understand this. Between the circular muscle coat and the submucosa, you have the submucosal plexus, which is also known as the Meissner's plexus. And between the circular muscle and the longitudinal muscle, you have the myenteric plexus, also known as Auerbach's plexus. Auerbach. I don't know how to pronounce it, but you get the point. Okay, so the circular muscle coat, which at certain regions such as the pylorus form a thickening to form the pyloric sphincter, and the longitudinal muscle coat, which essentially helps in the transmission of the wave of peristalsis. In certain regions like the large intestine, it undergoes modifications to form the tinea coli. And finally, you've got the last layer, that is the serosa. Now, if it is an organ to which the visceral peritoneum is attached, then you have a mesothelial lining. Otherwise, if it's a bare area where there is no peritoneum, it's just going to be a connective tissue layer with mostly collagen fibers. Okay, so from within outwards, you have the mucosa with lining epithelium, lamina propria, muscularis mucosae, then the submucosa with some submucous glands, then the muscularis externa with the circular and the longitudinal layers, in between both of which you have the myenteric plexus, and between the submucosa and the circular muscle you have the submucosal or Meissner's plexus, and finally the serosa / adventitia. If peritoneum, then serosa. If no peritoneum, then adventitia. Okay, now let's see what exactly is happening as we traverse from the mouth to the anus. This is stratified squamous epithelium, and it should immediately point towards esophagus, because there is no absorption taking place in the esophagus. The esophagus is merely passing the bolus from the mouth to the stomach, and when you're passing a bolus, you do not want a lot of friction, so you've got multiple layers, and that is one of the most important sites where stratified squamous epithelium is seen. Correct? As you go from the esophagus to the stomach, see what's happening? These are these tall columnar cells, which are the cells of the gastric mucosa. Okay? Moving back here, the lamina propria will show a couple of aggregation of lymphoid follicles and a couple of capillaries, which are going to be the capillaries in the lamina propria. But when you go here, see what's happening? The mucosa dips inside. You see, these are all dips, which are obviously known as crypts, and what is around these is actually what is known as the gastric glands. Okay, so the gastric glands, they are lined by three types of cells. Okay, you obviously have the common mucous neck cells, but the other ones that we're really interested in are the parietal cell, which is responsible for formation of hydrochloric acid. It has a very typical structure, the details of which I won't go into, but remember, write it down: parietal cell. Then you have the cells that are going to secrete pepsinogen, correct? The peptic cells. right? And you have what is known as a neuroendocrine cell. Now, you can read about it if you're interested. Neuroendocrine cells are present all throughout the GIT, later in the small intestine and large intestine as well. And there are special type of cells whose granules are not facing the apical side but the basal side. So it's very interesting because the rough endoplasmic reticulum is more concentrated below the nucleus. It's known as infranuclear RER and infranuclear Golgi, because the secretions of an neuroendocrine cell, also known as an APUD cell, are going to be secreted not into the lumen but into the capillaries that lie in the lamina propria. Anyways, I divert here, but it's very interesting. You should read about APOD cells. There's a separate tumor known as neuroendocrine cell tumor, which is very interesting. Read about it if you can. Write that down. APUD cell / neuroendocrine cell. So anyways, gastric glands and the different types of cells found in the gastric mucosa. Okay, now I'm going to go down again and we're going to traverse towards the duodenum. Okay, the stomach of course has a slight variation in the fundic area and the pyloric area. There's a couple of differences. There's a table in your textbook, read about it. Let's move on to the duodenum. Now what's happening in the duodenum is that the cells of the duodenum, which are known as the cells of the small intestine, also known as the enterocyte, is a very interesting cell because it has microvilli at the apical end for increased area of surface absorption. Embedded in these microvilli, coming out as bristles of a brush, are certain carbohydrate chains which are important because they kind of form like a protective layer, thus protecting the gastric as well as the intestinal mucosa. Then the lining epithelium, just underneath that, in the lamina propria, you're going to have all your capillaries. And then in the submucosa, you see this B? In the submucosa, you're going to have what are known as Brunner's or Brunner's glands. Okay, the Brunner's glands are responsible for secreting bicarbonate-rich secretions. Now, you need to understand that physiologically speaking, the pH in the stomach is more acidic because of HCl, and the pH in the duodenum and most of the small intestine is alkaline. So where is this alkalinity coming from? This alkalinity is coming from the Brunner's glands as well as pancreatic exocrine secretions. The reason is the enzymes that will be active in the stomach region and the enzymes that will be active in the small intestinal region are slightly different, and they get activated in acidic and alkaline pH respectively. So try to understand what's really happening histologically from a physiological point of view, as I'm saying. Okay, now we've reached the terminal end of the small intestine here, which is the ileum, and that's characterized by, you see this box, that's basically encapsulating a nice purple lymphoid follicle, and these are a characteristic feature of ileum, and these are known as Peyer's patches. Okay, please write that down. Peyer's patches. Very interesting things. They are actually let's just call them a bunch of policemen that are waiting just underneath the lamina propria to catch any kind of fishy activity, right? Suppose there's a Ganpati procession going on and people are doing Visarjan and everyone's getting drunk, and then there's going to be certain regions where the processions take place where there's going to be little cops who are going to be standing together to catch somebody who's going to be drunk and misbehave. You know what I'm saying? That's very similar to that. It's very similar to that. It's very amazing how the immunity functions work in the gastrointestinal tract. They're known as GALT. By the way, they're also known as MALT, mucosa associated lymphoid tissue. Anyways, that's a separate topic. As you go towards the large intestine, can you see these white little things? You know what these are? These are known as goblet cells, right? And the goblet cells essentially are white because the mucopolysaccharides are lost in dehydration during tissue processing, and so there's just like a ghost cell that is seen over here. Now they already start appearing towards the terminal ileum, and the number increases as you go towards the large intestine. Everything else in the large intestine pretty much remains the same, but the number of goblet cells start increasing. You can again see a lymphoid aggregation here and the large intestine, right? So as you just observe, the lining epithelium is changing as you're going from the esophagus all the way down, right? It's different in different places. I want you to make a note of that, how it differs and why it differs. The muscular part as well, because in stomach you've got an extra layer of muscle, the oblique layer, so you remember that. The submucosa, like I said, in esophagus and in duodenum is showing glands. In the ileal region, the submucosa, as well as the lamina propria for that matter, is showing lymphoid aggregations which are known as Peyer's patches. Now these are just a couple of features, but just try to familiarize yourself with some of these other things and try to apply yourselves while doing so. Okay? Alright. Now I'm going to go to the endocrine tissue that is seen in the pancreas, and this is known as the islets of Langerhans. Pale staining islands lying in between the pancreatic acini, and these are known as islets of Langerhans. Now before I even begin, I want to speak a little bit about the endocrine tissue. Here are some of the characteristic features of endocrine tissue. It's almost always capsulated. There's always a capsule, and the capsule always sends in little fibrous septa inside, and these are the fibrous septa that divide the cells that are responsible for the hormone production, into separate clusters or cords. Okay, primarily in case of the islets of Langerhans, this capsule is formed by reticulin fibers. Okay, that's a characteristic feature. And whenever you think of the islets of Langerhans, I want you to think of a pomegranate. So I don't know if it's pomegranate season right now, but whenever it is, open the pomegranate and you'll see the yellow colored septa of the pomegranate going inside, right? These are the fibrous septa, and then every little red colored pomegranate thing, whatever that is called, can be considered to be cords of cells and clusters. Now in case of the islets of pancreas, the cells that are in the periphery are mostly stained by acid fusion, and these are mostly alpha cells. So you can't see that because this is H&E, but the alpha cells are in the periphery. About 20 to 25% of an islet peripherally is constituted by the alpha cells. Physiologically speaking, these are responsible for secretion of glucagon, can be known as the expending hormone. So every time you stop eating and your insulin goes down, your glucagon goes up, and that is secreted by these alpha cells, right? In the center, you mostly have cells which are known as beta cells. They form about 70% of the islet constitution, and the beta cells are responsible for secreting insulin, which is the building hormone, correct? And there's a couple of other cells randomly interspersed, mostly again in the periphery, and these are known as delta cells, which are responsible for secretion of somatostatin and also gastrin to a certain extent. Now, one of the most important ways of identifying an islet is just look for pale areas when you're going through the slide of the pancreas, correct? And then if they ask you in detail about the structure of an islet, start from the outside, say that there's a capsule, the capsule sends in fibrous septa, and then there are cords of cells, or there are clusters of cells which can only be identified by using acid and aldehyde fusion. The acid fusion will stain the alpha cells, the aldehyde fusion will stain the beta cells. The delta cells are actually stained by argentaphin, so that kind of does belong to the whole neuroendocrine system or APUD system of cells, and it is stained by silver salts, which is a characteristic feature of all of these cells, by the way. Hence they're also known as argentaphin cells, argentum meaning silver. Remember this, if you use special stains, in case of this slide we've got acid fusion, which is staining the alpha cells with some pink. Can you see that? These are the alpha cells, and towards the center you mostly have the beta cells. Okay, what I really want you to remember about any endocrine organ, for that matter, is the fact that it has fenestrated capillaries. Now think of it this way: an exocrine gland has a duct system to pour its secretions wherever it has to. The endocrine glands have to pour their secretions directly in blood, and the hormones have to act at faraway places. So the only way that can happen is if they have a rich blood supply. So all endocrine organs are heavily perfused and they have fenestrated capillaries, so the hormones can immediately mix in the blood and create the effect that you desire it to cause. Okay, so remember that. Please read a little more about features of endocrine organs, and then you can start applying these facts to other endocrine organs that you shall study. You'll see that the suprarenal has pretty much similar structure. We're going to use special stains for chromophils and chromophobes and whatnot. The thyroid you'd probably have to study a little more about the follicular cell and how it is cuboidal. It can be low cuboidal or high cuboidal depending on activity, as I mentioned before. But just remember that now you have a basic pro forma of some of the things that you might find yourself repeating when you read something else that is closely associated. Okay, so that that pretty much sums up my lecture. I think we're running out of time, so I'm just going to quickly summarize everything. Now I want you to forgive me if you were expecting something very elaborate about every tissue and every slide in detail, because this is not the time to do that. And how will I know which subjects to choose? Like, if you could interact with me and tell me if there's a certain microscopic tissue or a histological phenomenon that you want me to talk about, I'll go ahead and make a video for that if and when I get the time. But for now, to summarize everything, the most important thing that you need to understand about histology is that every cell has a certain structure for a reason. And the only way to find out why the structure would be the way it is to understand the physiology that is happening inside the cell. Is it actively secreting something? Is it not? What is the shape of the cell? Is it tall? Okay, why is it tall? Is it short? Okay, why is it short? Does it have any kind of granules? Oh, great. Why does it have these granules? Who is creating these granules? If it has granules, are they basophilic? Are they acidophilic? Do they need any special stains to identify the granules? Then when you start thinking about tissues, that is group of cells that come together to form a tissue, it's always important to remember what the function of the tissue would be like. If the function of the tissue would be to prevent friction, you would go more towards like a stratified epithelium with like a dense connective tissue underneath it. If the function of the tissue is more absorptive, you would probably be having cells with microvilli at the apical surface. If the function of the tissue is more to put granules in the blood system but not in the lumen, there would be infranuclear Golgi, infranuclear of endoplasmic reticulum, so on and so forth. If the function of the tissue is going to be conduction, it's going to have really long protoplasmic processes, which is a feature of axons. But anyways, we're not talking about neuro. We don't talk about neuro in such situations. Organ. Now, technically the organ is when different kinds of tissues harmoniously live together to create a nice function, right? So you saw it beautifully in gastrointestinal tract, how the lining epithelium, the lamina propria, the submucosa and the muscularis move together to create a smooth functioning of the gastrointestinal system. Thank you so much for having me. I hope this kind of gives you a little more perspective on how to approach histology. And please let me know how you found this. Any kind of feedback would be very valuable so I can improve my teaching style and get better at what I do. Thank you so much for listening. All the best for your exams. Here's hoping that they get postponed. Where's the camera? Where's the camera? Here's hoping that they get postponed. Study well, and whenever you study, don't think about the exams. Think about knowledge, not information. Good night. Bye.